explain it in a way that is easy to understand. So I thought, it's not fair that I'm the only one taking advantage of his time and learning these concepts from him. So I thought, why not have a bigger audience? So we all can learn about a few things from him altogether. So that is why this talk has come around. We will try to keep it informal. So if you have questions, feel free to message. So over to Ani.
So thank you, Amit, for this wonderful introduction and for everyone who's attending this. And before I begin talking about the subject, Amit, of course, has been a pleasure to talk to. And I think I really like to talk to an audience that is engaged. And I've always been interested in the process of discovery. And I think it was Bill Bryson, the enormously gifted travel writer, who said about someone, I'm paraphrasing, that and he was describing some character, that he had the curiosity of a person who wanted to know everything there is to know about the world and explain it to everyone in it. And I do consider myself somewhat like that. So this is an experiment that Amit and I sort of talked about when he was visiting, where I'll attempt to convey some interesting ideas and concepts that have been fascinating me. And uh, the topic of today is diabetes, and I must say that this is, of course, not an advisory on what you should do and what you shouldn't. That's not my expertise or job. But this is a description, more or less, on how we systematically unpeel the layers of this extremely complex disease and how we got a better understanding of it. And then, more interestingly, how extraordinary people, scientists who were driven by curiosity and the desire to make a difference, got us to revolutionary treatments that have transformed the world. So I'll start sharing this. I've made some slides here, and let me share those with you. Okay. Amit, all good?
Yep, all good.
Okay. All right. So the first thing is that the reason we need to worry about diabetes, as everyone who's on the panel already knows, is that one of the biggest problems of humankind. It's a disease that kills a lot of people, and if not just kills, it's a big problem. It leads to many health issues that we will hear about in the next part of the talk. About 31 million people in the United States and about 70 million in India. And of course, if you live in India, you know this. That this is one of the biggest health problems in India. There are many people who remain undiagnosed, but even the diagnosed ones, it's believed that in the next 10, 15 years, there'll be 100 million people in India alone that have this disease. And of course, there is the growing incidence of diabetes also remains a problem.
So, Ani, is this something which is caused only by a modern lifestyle, or has it been around for longer?
That's a very good question, actually. I think the answer is, of course, both. It is not a new disease by any means. So let's understand what is the disease and have people already known about it. The very interesting thing when I started reading about this a few years ago now is that ancient cultures actually already knew about this disease, and it's really fascinating to hear or read what they found. Because remember that in those times there were, of course, many diseases. Most of them were not really very well characterized, so there were people who systematically went and looked at patients that had specific similar symptoms and clubbed them together, and then realized that there was a group of patients that had this very curious disease. And so this action actually, and this is also a point of interest, happens in the area around the Middle East and India and China and Northern Africa in Egypt. So this is where most of the research, which is, of course, it's going to be called research today. It was a systematic interrogation and investigation of this disease, and it was done. So the first description, some of the earliest descriptions of this disease, is from what is called the Ebers papyrus. So this was a German Egyptologist, George Ebers, who was very interested in diabetology, and he went and bought these or acquired this Ebers papyrus from Egypt, and then it got translated. And if you see this, it's very fascinating actually. How if you look at this, there is a description of a patient which is a clear description of a disease that looks like diabetes. If you examine someone sick in the center of his being, and this is also very fun actually, if you like this, to see how people have looked at these documents and translated them. So there was this description of not one but a couple of patients at least in this, where they said that these patients had excessive thirst that was unquenchable and frequent urination. And what they described is that these patients would waste eventually and unfortunately die. And so this is believed by many people to be a clear description of diabetes, although it is a little bit contested. This was in 1550 BC. So you see that millennia ago, actually, diabetes had been described already. Now we come to the 6th century BC, where Sushrut, who is known as the father of surgery in India, has this incredible document or book, which is called the Sushrut Samhita. And in Sushrut Samhita, Sushrut has actually a very good description of diabetes, and is astonishing the kind of descriptions Sushrut makes of patients, again, who have excessive unquenchable thirst, and it's a thirst that cannot be quenched. And Sushrut says that they keep consuming water and then losing water through urine. Now the interesting thing that Sushrut notes is that the urine is very sugary, and the the very interesting thing that has been written about quite a bit is that Sushrut noticed that the urine of the patients who have this disease, where they have excessive thirst, excessive urination, the urine is sweet. And how do they know that? This is a recurring theme in the talk today. And throughout the ancient and medieval ages, people would actually taste the urine of the patients. And they found that the urine was actually sweet of these patients, and it had a lot of sugar. And Sushrut notes that the urine of patients with this particular curious affliction would attract insects and ants, and so calls it Madhu Meha. This is an astonishing insight. This is called Madhu Meha. Madhu in Sanskrit means honey, or it also means sweet. And Meha is anything that has to do with urine. This is a description of Prameha, or all the diseases that have to do with urination problems. And then in 2nd century AD, Artias of Cappadocia, who was a Greco-Roman individual, he was uh, considered to be a very influential person and had written a treatise on medicine, has this really wonderful description again of many diseases, including cancers and ulcers and kidney disorders. And then he has this description of a disease that if it matches the description, and of course, it doesn't have a name yet. So he decides to name it diabetes, which basically means to siphon or to pass through, which is again referring to the fact about the problem that he also observed about urination issues in uncontrolled diabetic patients. And this is a Greek word that still stays. Now, when I was reading this, I was also astonished to find the works of Ibn Sina, who is also called Avicenna by the Europeans. The work of Ibn Sina, who was a Persian but worked with many different Persian kings, was very interested in medicine, collected the works from many different cultures, including the Chinese cultures and Indian and the Greek medical books, translated them, and wrote this book called the Al Qanun Fit Al Tib. So, for interested people, Al Qanun, Qanun is an Arabic word. This book was written in Arabic, and it's when it was translated to Latin, it got called the Canon of Medicine. So, this is an interesting aside. This word still exists and is used in India and in many places. Canon in the Latin inspired world, and Qanun in our part of the world. But what
So,
was very,
Adi,
yes.
if I could just jump in, so were these people like in Egypt, India, were they speaking with each other?
They are, so if you notice, these are very different times. Okay, they are
Yeah,
separated
yeah.
sometimes by centuries. But to your point, the interesting thing was that yes, the books were being exchanged. They were reading each other's books. Sushrut Samhit, for example, was translated into Arabic, and it was called Al Kitab Sushrut or something like that. So yes, there was definitely They were reading each other's books, translating each other's books from different languages, and that is what actually did inspire. It's basically like right now, right? That you would read journals, reference them, and carry that work forward, and that's what was going on, which is fascinating if you think about it.
Yeah, and even though there are these are different parts of the world, and there's no modern lifestyle yet the disease of diabetes is being observed at all these different places of the world.
That's absolutely true. Yes, and this is fascinating that many different people in different parts of the world were making observations that were very similar to each other. All right. So then the action moves from here, from Asia, from India, China, and the Middle East to Europe. And there's an interesting point to be made here is that when we read about discoveries that people were making, it is always interesting to see that there is a clear link between economic stability, prosperity, and just the absence of strife or war. And then when that happens, the society has made some of the most astonishing discoveries, and that's a point that I'll come back to later in the story of diabetes. So English doctor Thomas Willis described in 1674. We are now in the 17th century, and again they were still using the same method. He was sipping from a glass the urine of a patient, which seems gross today. That was the standard of the state of the art at that time, and says that the urine is wonderfully sweet, as if it were imbued with honey or sugar. And then another English doctor, what they do is that he boils the urine of this individual, sees that there is a crystalline residue that tastes like brown sugar. This is also interestingly an observation that Shushrud and Ibn Sina had independently made, saying that if you leave the urine out to the and expose it to the elements, you find that it eventually starts getting sticky, and then sticky to the fingers, and then it leaves a residue that almost looks and tastes like sugar. So you see how interesting this is. And so Thomas Willis decided to add the word mellitus to it, which is a Latin word for honey. And so you can see that what diabetes mellitus actually means is not very different from the word madhumeha. So the big ideas here were that sugar, they noticed, appears in and disappears from the blood, and especially the urine of some people that have this affliction. And this is a specific disease that whose at least overt symptoms that people could see are excessive, unquenchable thirst, frequent desire to urinate, and then eventually and unfortunately wasting and death. And what was noted also by Sushruth, which is again very interesting, is that he noted that this disease appears to be linked with the consumption of food. And it is astonishing that Sushruth writes that these patients with this symptom are often in the higher castes, and they have access to what he calls rich food. This is very interesting because he has already noted that this is not a disease of of a deficiency of something, but rather an excess of something. And then there are, of course, recommendations that they should stay away from food grains and others. And not all of them are correct by modern understanding of the disease, but it is still astonishing that in 600 BC, um, these interesting principles were already established.
So one question then I have obviously is that everybody is observing that your urine is sweet, something is going on. But then how do you go about finding out where in the body is it coming from? Like the body has so many different organs, so many things going on. How do we go from this observation that urine is sweet to the cause of that sweetness?
That's interesting, Amit. And I sort of almost expected that question from you coming, with a background in engineering. Of course, once you know that there is a disease with specific symptoms, the clear question that comes to your mind is what is the site at which this is happening? What is the organ that is involved in this? And so this was the question that, of course, people had. Now, I remember I told you that now the center of action as in terms of science has moved to Europe, And that's where people are starting to ask these very interesting questions. And before we start thinking about this, I just want to put in your head the idea that if you want to know which organ is or what function of what organ is disrupted or impaired in a disease, you want to know the various parts of the disease of the body. So imagine that you have a car which is not working very well. The first thing you would want to know is what are the different parts of the car. You would take the car apart and see that there are different. There's a spark plug, there's the engine, there's the steering wheel, there's the tires and stuff like that. But just cataloging the parts will not tell you the answer, because you have to also understand what is the link between those organs and how they work together to make the car function. And only that can tell you how the car, uh, why the car is not working the way it should. And the reason I bring this analogy up is that in the initial days, most of the studies were in anatomy, so which is the understanding of the parts of the body, but not physiology, which is the understanding of how these parts function together to in a living organism. And this is a very important inflection point in the history of medicine. So when people wanted to study human anatomy, of course, there are all these problems that you have to dissect human corpses. So in the early days, human corpses could be dissected, but then there were religious diktats that prevented people from using human corpses. And so people then started to use animal corpses. And again, a point to be made is that the only reason why many of the therapies that we use work is because animals, at least some of the animals, are so very similar in their anatomy to humans. And but still, people did realize that we need human anatomy studies. The regulations were not as strict from the governments, of course, but the regulations from religion where there were diktats that you cannot do this and you cannot do that. And so there are very interesting stories when people started to even raiding graves to actually take out corpses because they were that interested in anatomical studies. So they had made a very nice distinction between the different organs and had written about what these organs do. So many of these were wrong. Many of these assertions were later found to be wrong, but it was a very good beginning. And so there, th- I need to mention Claude Bernard, who was a French scientist, a very interesting person. He he did medicine, but he was interested more in research, and this is not really common at that time. Uh, in the 1800s. And so he, as soon as he finished his medicine, he went into a research lab, and he says that I consider the hospital the antechamber of scientific medicine. It is the first place where the physician makes his observations, but the laboratory is the temple of the science of medicine. So you see that this is the beginning. Now we know many physicians, scientists, and this seems to be very common now. But he was one of the first who said that we need to learn medicine, but then get into the labs. And he was interested in understanding the links between organs and their function. And he got married. The story goes that he was not a very well-to-do individual. He got married through an arranged marriage, and his wife was actually very well-to-do. And so it is it is said that he used a lot of that money for research. That's an interesting. Now the really interesting thing here is that his wife was really interested in animals and dogs in particular. And she realized that he also had a fascination for dogs. And so I think it started very well, their marriage, but then she soon realized that his interest in dogs was actually very different from hers. He wanted to know what happens, for example, if you feed sugar to a dog and kill the dog immediately to see what is happening to the sugar. So he would do all these experiments. He started doing experiments in dogs. In fact, dogs are going to appear in the story many times. You will see the importance of dogs in this whole story. But he started doing these experiments. Of course, his wife was aghast. And
Yeah, so science is very different now. I'm pretty sure such experiments will not be allowed today. But so
absolutely.
much of so much of what we have learned about science is from the wild west of science, when people did these crazy experiments, sometimes with themselves and sometimes with unfortunate other people or
That
animals.
we could do a completely separate story on that. That's a fascinating topic. But yes, this none of this would be allowed. Of course, you know that we all do animal experiments these days, but they are very tightly regulated as they should. And yes, you are right in that many of the discoveries did come from the Wild West. People doing experiments that they should not have. And so what he did is that he realized that there was a connection between the food we eat and the different organs. He did believe that the food we eat is broken down in the gut, and then there is something that is happening in either the lungs or the liver or the pancreas. And of course, he did not actually suspect the pancreas to be doing anything, but the location of the pancreas, this organ that I show you here, that is very close to the small intestine or the intestine, and it has these connections, these ducts that go in, made him wonder that the pancreas was playing some really interesting role. And he has done some really cool experiments that I, won't explain in detail. But he saw, for example, that if you look at uh, fluid above the pancreas and below the pancreas, there is a very big difference in the clarity of the fluid, suggesting that the pancreas is putting in some juices into the intestine that are clearing or digesting the food. So this was a very cool demonstration. But the big demonstration and the big finding from Claude Bernard was that he found that sugar is very tightly regulated in the body. And this was a very important discovery. And then he said that I think that this regulation of sugar uh, is destroyed in diabetes. And I think that was, remember, that was not known at that time, the link between food intake, how it gets converted into sugar, and how the sugar increase in the blood is linked to diabetes. That was not yet known. And Claude Bernard's work, he's of course done phenomenal work that goes above and beyond this. But this understanding of physiology, he's called the father of physiology, was key. So these were the big ideas. Disturbances in sugar control lead to diabetes, and he believed, unfortunately, and this is again a story that all of these scientists, starting from Sushruth to Claude Bernard to Ibn Sina, they made astonishing discoveries, but they were right and incredibly right in some of the things, but they were also wrong. We know now demonstrably in many other things. And Claude Bernard believed that diabetes is a disease of the lung. He believed that. Uh, Food we eat is broken down into sugar, and the burning takes place in the lung, so it's a disease of the lung, which turned out later on upon experiment to be wrong. He realized that the lung was not the site, and he wrote in his notebook that I don't think I understand anything anymore. Which is very unfortunate because he was such a brilliant scientist. Some people did have a hunch that it was not the lung, it was not even the liver, it was something else that was playing the big role in diabetes. The reason they knew that is that there would be patients that would come into clinics, and they would have clear signs of diabetes. And, sorry, not patients, there would be people who died of diabetes. And when they would be autopsied, the physician or the would notice that they had an atrophied pancreas. They had a pancreas that had disintegrated or had tumors or had some sort of a abnormality, and they suspected that why is it that many patients with diabetes, when we autopsy them after death, we see that the pancreas has a big problem. Is there a link between the pancreas and diabetes? So this led to a hypothesis that what if you could just take the pancreas out from a living person. Of course, you can't do it from a person, but uh, from an animal, and see what happens. This required extraordinary surgical skills because this is again the 1800s, and it was not possible to take the pancreas out very efficiently and still keep the animal alive. That's where Oscar Minkowski comes in, this Lithuanian Jewish immigrant who was working in Strasbourg, which was at that time in Germany. And Oscar Minkowski with Joseph von Mering did this experiment. He was an extraordinary surgeon. So he said, I can do this. I can solve this problem. I can remove the pancreas from a dog, and I can see what happens. I can keep the dog alive. He saw that when he did that, his assistants would bring little dogs in, and he would cut them open, remove the pancreas, sew them back up. And then what he would notice, really very interestingly, is that these dogs would start urinating all over the floor. And he really would get mad at these assistants. Why didn't you bring a trained dog? And the assistants would tell him, "This is a trained dog. I don't know why he's doing that." And Oscar then realized, oh, this probably is diabetes. He went, he picked up collected the urine from the floor with a pipette, checked the sugar, and lo and behold, the sugar was really high. And he saw this was the first time that the disease of diabetes was linked to the pancreas. And Oscar Minkowski plays a big role here.
And unfortunately, also many innocent dogs.
That's correct. And an interesting aside, of course, is Joseph von Mering he thought the problem was solved, and he said, I'm not interested in this problem anymore. He went on to do other things, and he also did clinical trials on paracetamol, which we use to this day. So you can just imagine how incredible the work of these people.
So after he connected pancreas and diabetes, he sort of did not pursue that further, and it left.
Well, there was a problem, and I'll come to that. Okay, so this is just the description showing that the pancreas is the site of sugar control. But Amit, having worked in a lab with me before, of course, you didn't work in a wet lab, but the idea is the same. Is that he did one more very interesting thing, and I think that was key to the what was coming next. He not only made the diabetes model, if you will, where he removed the pancreas of a dog. He also wondered what would happen because these dogs are going to die in the next few days. They don't have a pancreas, their blood sugar is out of control. So he wondered what would happen if he took the pancreas of another dog and just implanted it in the skin of this dog. He did that, Oscar Minkowski did. And he saw that the blood sugar now was restored to near normal levels. And only when, because this is an implantation that's imperfect, the artificial or the foreign pancreas is going to be degraded in a little bit. And so when that happens after a few days, the dog would then still die because the foreign pancreas is not functioning anymore. But this was a clear demonstration that the pancreas is not just necessary but also sufficient, the kind of experiments we do in the labs these days. And it was really astonishing to see how he did that in 18, I forget the year, but it was in the 1800s. Now, here comes in another scientist, the German Langerhans. And so, what Langerhans showed, because now we are zooming in into the pancreas, but the pancreas is not one monolithic organ. It has different types of cells. In fact, the pancreas has different areas in the organ that do different things. So, and his Langerhans superpower, if you will, was that he
Yeah,
had learned from his boss how to make nice little sections of or cuts of organs and stain them with colors to look at them.
yeah. I think that can be said in general about biology. Every time you think you know something, there's like five more levels of complexity.
Of course, and that's
Uh,
that, as you know, I mean, that is the problem why most of the diseases, because of the complexities involved, have been not been able we've not been able to find cures, treatments at best, but cures take much longer. And this is a theme that we will be using recurringly throughout this 2 or 3 part session. So what Langerhans said is that in the pancreas there are little areas that look like islands, and these became known as the islets of Langerhans. Uh, uh, Taking sort of his name. And you see here in this staining that these are these little areas that have cells that look very different from the other cells around it. And I was talking to Pam about this the other day. The interesting thing here is that these islands and the cells that make up these islands and the cells that are important for the rest of our story actually make up only 1 or 2% of the entire pancreas. This is a very important point, is that the most of the cells in the pancreas are actually not involved in the function that we are going to talk about, which is the production of this little magical thing that people started hypothesizing or talking about, that there is something in these islands that is being made that is important to the story of diabetes. And so it was called because it was the made in these islets or these little islands. And the word for that is insula in Latin. It was called insulin, which, as you, it's again an interesting aside that how uh, you've used this word insular, insulate, isolate. All of these words come from the same root, insula in Latin. So the big ideas here were that the work from Oscar Minkowski found that pancreas is the site of the action. Some trouble in the pancreas can cause diabetes, and restoring, this is important, function can reverse diabetes. And Langerhans, the islets seem to be the site of production of this mythical magical thing, which was called insulin after insula. And then the big story here is that people realize that isolating this thing, insulin in pure form, may help reverse diabetes. So you see how we come all the way from the description of the disease to zoning in on the pancreas, to the cells in the pancreas that are important, to the actual uh, actual element or the chemical in those cells that is important for function and for reversing the disease that was seen in these patients.
Yeah, that's a great point. So, for thousands of years in different parts of the world, we observe the sweet urine. And then, when the modern science started, due to some unethical experiments, we now narrow down the organ and then we narrow down to some cells. But then, how does one find a cure? Like, okay, how do I stop it? I know where it's coming from. How does that process start?
So this is a very interesting question, Amit, and of course it begins with insulin. And the big problem with insulin or trying to isolate insulin had to do with the pancreas. So let's go back to look at the pancreas. So the pancreas is this organ. This is this large organ that is shown here. And as I mentioned, the islets are very few. It's 1 to 2% of the organ. So most of the cells in the pancreas actually have what is called an exocrine function. So what they do is that they make these digestive juices that are emptied into the gut, and they are important for digestion. And the islets are the regions in the pancreas that have what is called an endocrine function. So they make hormones, and insulin now we know is just one of them, and that's going to be interesting for our next stories. But insulin is one of the hormones that is made by the cells in these islands. So what the people in the early days would try to do is that they would try to take the pancreas, they would try to mash it up, take all the juices and try to inject it into the diabetic dogs, and they barely saw any effects. And the reason was, and this was an astonishing finding, and this is what Banting and others, who I'm going to come to in a minute, thought, is that what if the reason that insulin is destroyed when you do this is because there are digestive juices that are being made by the rest of the pancreas. So when you mash the pancreas together, you actually, the digestive juices that are being made by the cells here are digesting the insulin, and so it's not active anymore. So the big engineering challenge was, how do you get the stuff from these cells, from the little islands, without having an effect of the other cells in the pancreas? So this was an interesting engineering challenge, and this is where we go to Canada, to Toronto. And again, the interesting point here is that the reason why most of the diabetes research action now shifts, I told you how it shifted from Asia to Europe, and then this is the 1920s, and World War I has started a few years ago, and so now Europe is in strife. So this action now shifts to North America. And this is again an interesting point about that I was making on how economic stability and just the absence of strife is linked to discoveries in general. So there was this surgeon, there was this physician, uh, um, Frederick Banting. And Frederick Banting is probably one of the most interesting people in this story. He is considered widely as the most famous Canadian, perhaps more than Justin Trudeau or Justin Bieber, uh, depending on whether you're a scientist or not, or whether you care more about science or not. But Banting read a paper on the islet of Langerhans and its importance to diabetes, and realized the problem that I was telling you. He realized that if you try to isolate the internal secretion, then you could relieve glycosuria, which is just a fancy word for glyco is sugar and urea is in the urine. So he said that if I can isolate this, I can try and cure the high sugar in these patients. So he thought that there was a clever trick that could be done. What he did, I have to go back to that slide here. What he did is he had learned a process where you could ligate or tie the duct, the pancreatic duct. So when you did that, when you shut off, tied that duct, what would happen first is that the cells, the digestion producing cells, the digestive enzyme producing cells would atrophy or die, and then that would let only the islet cells remain. And of course you had to do this really carefully. You had to do this with deftness, and the timing was very important. And just learning that, as Banting did, uh, really changed the world, as you will see. What Banting did is he tried again to do this in dogs. I think in the story of diabetes, dogs were just the model of choice because of the early experiments from Claude Bernard and Oscar Minkowski. By the way, coming back to Claude Bernard, who initially started doing all these experiments in dogs, he he kept working on dogs, and as you can imagine, his wife, who was a lover of dogs, did not really appreciate that. They got a divorce, and she became one of the biggest animal activists, leading sort of to the birth of animal rights activism and tight regulation of work in animals. Amit, to your point. But returning to Frederick Banting, so this is a dog. I think this dog is Marjorie. This is the dog. What they did is that, and this is uh, an associate to Charles Best. So what they did in this dog is that they repeated Oscar Minkowski's experiment. They removed the pancreas. They saw that the dog would get diabetes, had high sugar. Now there were better methods to look at sugar through acid hydrolysis method. You didn't have to taste it, so you could actually look at how much sugar there is in the urine of the dog and also in the blood of the dog. So they knew they had recreated this model. And just a point that if you want to cure any disease, as we all try to do in the labs, we have to rely on an animal model that reflects the patients and the disease that we are trying to cure. So this is just for the non-scientists in the audience. The importance of that cannot be understated.
Yeah, and in modern medicine, we try to be as ethical as we can when using animals.
Absolutely, absolutely. We use the fewest numbers of animals. We use them in a very ethical manner, and there are many rules that disallow the experiments that they did in this. And there are other methods, and this is a subject matter for another talk, where you actually can even get away from the use of animals. And there are many different really cool technologies that are emerging. But back to Banting. So Banting realized that if I create this dog model of diabetes, the dog is going to die. But if I now purify insulin from the pancreas of the dog and from the islets by tying the duct, I may be able to keep this dog alive. So, this is Marjorie, a dog that was injected several times with this magical potion that Banting isolated through his deft skills together with Charles Best. And the dog remained alive for up to 70 days. And this was really considered miraculous.
So,
And
from where was Banting deriving the insulin? From what? Was it other dogs?
it was from dogs initially, but then it was from pigs because he
I
realized that
see.
he could just get more insulin from pigs.
So he
Later
could get,
on,
yeah, so he could get isolate insulin from the pancreas physically from pigs and then
yes,
use that to keep Marjorie alive for 70 days without a
yes.
pancreas.
Yes, so what they would do is that they would cut open the animal, they would tie the duct, wait for the digestive cells in the pancreas to die, and then take the pancreas and use some sort of alcohol and other extraction methods to remove and purify this substance. Again, we now know that it was very impure, but that was a great start. He saw that when you keep injecting them the dog can be kept alive. This was a big discovery, and Frederick Banting went and talked about this. He was very excited, but he didn't have a lot of resources to he and what is said is that he was a clinician, but he was not a scientist. So he went to, and I'll tell you why that distinction matters. I of course think he's a scientist, the one, if not one of the greatest. But this is a thing that he went to John MacLeod at the University of Toronto, who was a researcher, had a big lab with lots of resources, and MacLeod didn't take him really seriously because he thought he was not a scientist and he didn't understand what he was doing. But he decided to give this person a try anyways. And there are many interesting stories on how Banting and MacLeod didn't agree with each other because MacLeod thought Banting was messy and he didn't do stuff well, and he had, when he started working in with MacLeod, he had dogs all over the place and urine all over the place, and he was never organized. MacLeod used to be really very angry at him. So, and also MacLeod thought that he was not doing this systematically. MacLeod thought that he needed a biochemist, and Frederick Banting thought that that was not necessary. But MacLeod was in the end turned out to be right, because uh, what happened was that there was a murmur in the community because of Frederick Banting's exposition and talking to people that maybe, I have a cure for diabetes. So they decided to try it on a 14-year-old individual, Leonard Thompson. And Leonard Thompson was a 14-year-old boy that I'll talk about in a minute. And they said, okay, why don't we try and actually do this injection and see what happens? Now, uh, he's MacLeod thought that the biochemist should be involved because they know better how to purify things. Banting disagreed, and then they really had a physical fight. It is said Banting and Collip, the biochemist that MacLeod brought in, and someone had to come and intervene. Anyhow, they've later published this paper. They got together, they worked well together later on. Published this paper, which probably one of the most famous papers in the history of diabetes, the preparation of pancreatic extracts containing insulin. And the uh, contributions of Collip are very interesting here and very important, because he could purify much better and much larger quantities of insulin that were very important to the story that I'm going to tell. But before that, I think we have a special guest here today. My colleague, Dr. Pam Itkin Ansari. She is a specialist. She has done research on diabetes for about 2 decades and more. And she has done research that has gone on to clinical trials. She was a member of the Juvenile Diabetes Foundation, and she has given a TEDx talk. So I'm jealous, Pam. And why don't you tell us why this was such a big thing, and why knowing what insulin does would change history, as it were?
Thank you, Ani. I'm very happy to be here. Like Amit, I'm a great admirer of your research and just your prowess in science. So thank you for inviting me. So before 1922, type 1 diabetes and even type 2 was a death sentence. And what you see in these hospital beds are children who will die in general within a year of being diagnosed. And why is that? And what's so important about the sugar in the bloodstream? Well, every cell in our body uses sugar as fuel and needs sugar to perform correctly. We also use proteins that are broken down and fats in our cells, but sugar is the most important. So the idea of finding that sugar was in the bloodstream and finding it was in the urine, the problem was that our cells in our body weren't taking the sugar in to use as fuel. And the discovery of insulin led to us finally understanding that insulin from the pancreas is the signal to all the cells in our body to open their sugar channels and remove insulin from the bloodstream, just like a car engine needs gasoline. Now, there are 2 reasons that it's dangerous if the sugar isn't removed from the bloodstream. one, the engines in our cells can't work, just like the engine in a car without gasoline. But also imagine a car owner who thinks, well, I'm having trouble getting the gas through the pump into the engine, but I'll carry around a lot of extra cans of gasoline in my car. Well, of course, that doesn't help the engine, but it's also very dangerous, as you know, right? Those cans of gasoline could blow up. And similarly, having too much sugar in the bloodstream is a twofold problem. Number one, the cells in the body are actually starving because they don't open the sugar channels without insulin. Number two, leaving all that sugar in the bloodstream, the sugar molecules get stuck places they don't belong. And that's what leads to all the complications of diabetes. That ruins nerves and blood vessels, among other tissues, kidneys. So I'm just going to tell you really briefly, most of the patients in the hospital here had type 1 diabetes. In type 1 diabetes, our own immune system attacks the insulin-producing cells in the pancreas. And these children who were diagnosed would generally die within a year. They cannot live without insulin. And even today, the therapy for a patient with type 1 diabetes is insulin. Now, type 2 diabetes, you're going to hear about in future talks in more detail. It begins a different way. It begins with the insulin-producing cells working fine, and the rest of the body isn't responding to the insulin signal. The result is the same. The cells in the body starve because they don't open the sugar channels, and the sugar in the bloodstream gets stuck where it doesn't belong. Many people with diabetes will be familiar with getting an HbA1c test when they see the doctor. It's actually a measure of how much sugar has gotten stuck on hemoglobin molecules. So Ani, I'll let you continue and see if there are more questions.
Thank you so much, Pam. This is wonderful, actually, the way I love the analogies that you give and the way you described it. And, to the point that, the reason also I got
So
Pam.
Ani, there's a question. There are a few questions. So one question is, what is the difference between type 1 and type 2? So
Okay.
Pam, if you could explain with your analogy,
Sure.
taking it further.
Okay, I will. So in type 1, it's what we call an autoimmune disease, like lupus and scleroderma and rheumatoid arthritis. Something goes awry, and in general, a child's immune system starts attacking its own body. And in each of those diseases, the immune system is attacking, in general, a certain organ. In diabetes, it's attacking the cells in the islet that make insulin in type 1 diabetes. And as I said, this leads to too much sugar in the bloodstream, starving cells, sugar getting stuck places. Type 2 diabetes begins with the insulin-producing cells are fine. They're making lots of insulin. And this is what's associated more with obesity. The molecules being signaled from too much fat in our body is blocking the signal of insulin. So insulin's being made and it's being ignored. But you can see that the outcome's the same. The cells in the body are starving, the tissues, the organs don't have sugar inside them to work like an engine in a car, and the sugar's staying in the bloodstream and getting stuck where it doesn't belong. Now, eventually, and Ani will tell you in a future episode about the wonder drugs now for treating type 2 diabetes. Treating type 1 requires we give them insulin. Sometimes treating type 2 can be handled more by blocking these aberrant signals that keep cells from listening to insulin. But eventually, if we don't fix the problem in type 2, the insulin-producing cells will get overworked and overtired. So about 25% of patients with type 2 diabetes eventually need insulin shots too, because their insulin-producing cells got exhausted by making more and more insulin that was being ignored.
Okay, that's a great answer. So in type 1 and type 2. Okay, makes sense.
Thank you so much, Pam. I think
one more question. If the digestive juices in the pancreas degrade the insulin in diabetes, why does this not happen in non-diabetics?
Oh,
Does it?
this is so brilliant. So
Yeah.
here's what happens. The digestive enzymes actually go into your intestines through a ductal system. And so they never touch the islets in your own pancreas. The insulin from the islets has lots of blood vessels around it, the islets. So the insulin doesn't go into the gut. The insulin goes into your bloodstream to tell every cell open the sugar channels. What happened though, when people tried to mash up the whole pancreas, they wanted to isolate insulin, but now they put those digestive enzymes in contact with insulin, which never happens in a normal body. So it's the organ is really, it's the pancreas, it's really like two separate organs. And the products go two separate places. They never touch each other normally. It was only difficult to purify insulin when people tried to mash up the whole pancreas, because all those 98% of the cells in the pancreas that make digestive enzymes now broke down all the proteins from the islets also.
So in the body, there's a very special equilibrium and everything is working separately. It's difficult to reproduce that in an outside system.
Yes, and it's actually very odd that those two things are in the same organ because they have such different functions.
Yeah, thankfully we don't have to depend on pig insulin anymore, and there are better synthetic ways.
Exactly.
I think this is a very important point, and I think I'm glad that this question was asked, because as Pam really beautifully explained, I just want to add a very interesting thing here, Pam. The fact that you very nicely showed how the separation of the digestive part that connects to the gut and the hormone producing part of the pancreas that connects to the blood. The example I want to give, this is a digression and we are going to be a little bit over time, but I'll very quickly mention there are many snakes which make venoms. And one of the things that people wonder is that how do these snakes actually not, how are they not affected by their own venoms? And one of the cool things that happens in some of the snakes is that they have different chemicals that only mix together when the snake bites the prey, and these two chemicals will come and mix together in the prey. So this is how they have a separation of those two chemicals, and the chemicals actually never mix together with each other in the host organism. It's such a fascinating way of looking at things. And
so one more question. Does physical exercise impact insulin in any way?
Okay, well, that is probably going to be discussed more later. But let me say, for a patient with type 1 diabetes that has an autoimmune disease, exercise is not involved. Now we talk about a patient with type 2 diabetes, where obesity and inactivity do play a role. Because as I mentioned, the fat tissue in the body is blocking insulin signaling to all the tissues to take up sugar. Well, exercise separately, even from losing weight, has beneficial effects on stopping the blocking of insulin signaling. So it's even important for people who don't lose weight to exercise. That can improve how the body responds to the insulin that their own cells are still making. And it brings up a very important point. A patient with type 1 diabetes has no way that they themselves might reverse or improve their disease. They need insulin. Their cells are being destroyed by the immune system. That doesn't mean it can't be cured eventually, but as the patient themselves has no power to do that. With type 2 diabetes, it's going to become clear that with changes in diet and exercise, many patients can reverse the disease, especially if they do it in the early times after being diagnosed. as I said, eventually, if nothing changes and it gets worse and worse, then their own cells that made insulin are going to get too exhausted.
Yes.
So, but yes, type 2 can be reversible, and that's very exciting.
Yeah, excellent. One more question. Are there any edible insulin yet?
I see that. Not yet, but, and I understand the reason for asking, because I know in my work on type 1 and type 2 diabetes, I've known many children, one I think of often named Calvin, who by the age of 6 had had 16,000 finger pricks to test his blood sugar and 10,000 insulin shots. So I really understand the question about edible insulin. It will probably come. There are many, many treatments and different formulations of insulin being made. With
So why is that a challenge? Why cannot we have like a insulin like a Tylenol or something?
well, the main problem with eating a protein, because insulin's a protein, is it goes through the digestive system. And we know what happens in the digestive system. Everything gets broken down,
Okay,
right? All
I see.
proteins. But I do think at some point it may be possible that there's a capsule or something to do this. There are also other kinds of ways of delivering insulin. Even now, instead of insulin injections, some people have insulin pumps. So there's a very, very, very thin threaded needle into their stomach area, not in the digestive system, just that area under the skin. And now they don't need an injection every time they want to pump more insulin in. So these are amazing advances that can be talked about in detail.
Makes
So
sense.
getting
So
easier all the time.
yeah, so a lot of the medicines we take, aspirin, paracetamol, they are really small molecules, so they get
They are
absorbed in
not
the bloodstream.
proteins.
Yeah, whereas insulin, it's a much bigger, 100,000 times bigger protein. And if we eat it, the body digests it, breaks into little parts. So we don't
Right.
get the benefits.
The darn
That's the
pancreas makes all these proteases that break down all
Yeah,
the proteins, so you have
yeah,
fuel, but they don't know not to break down insulin if you swallow it.
makes sense.
Mhm.
In fact, what you're trying to do is, when you ingest insulin, you're trying to mix together what our body intended to separate in the first place, right?
Yeah.
So you're mixing them with the digestive juices. So I'll just very quickly move on. We are going to be a little bit over time, but
Okay.
Pam, this is so insightful. This is wonderful information. We are all learning a lot. I know that you've worked with these children who have these enormous problem of even with the treatments that we have currently, which we describe as miraculous, there are still so many problems that remain, and we will talk about them. So now coming back to Leonard Thompson, who was the child that I talked to you about, there's a beautiful description of this in the book, The Drug Hunters and the 10 Best Drugs of All Times. And this talks about Leonard Thompson was a kid who was almost about to die. Again, this is 15th December 1922. I want you to pay attention to the date here and also the weight. Terrible wasting, 15 pounds, just 15 pounds. Uh, and the mother is seen carrying this very frail child. It's such a sad uh, sight. And Banting decides to try his injection of insulin on this kid uh, on 11th February 1923. Okay, so 2 months, just 2 months after this, and the injection doesn't work very well. It almost fails, but they do see a little bit of a sugar drop, encouraging them to do it again the week after. Colip, the biochemist that I told you about, says that I can get better insulin. I'm a biochemist, I know my stuff. So he gets this purer form of insulin. He does it from cows or pigs. I think he does it from cows, and he realizes how to make it purer. Does Banting wants to do the injection himself? He thinks it is his big discovery, but he's not allowed to because he's not qualified. So it's very interesting that they say, okay, you can take the syringe to the doctor, and the doctor can inject it, and this sort of massages his ego a little bit, and he goes in and does that. The injection is given on 23rd of February 1922. It's very interesting that it is almost exactly 102 years And 2 years ago, I remember there was a very big sort of celebration in Toronto of this. And this is Leonard uh, uh, Thompson. I don't know if I'm getting the name correct, but on 15th February 1923. So just look at the date. It's 2 months after, and look at the weight. It's 29 pounds. So the patient has doubled his weight, and is this is a triumph, and this is called a miracle. The news spreads like wildfire in all the community in Northern America and Europe and elsewhere, and people start talking about this everywhere. The interesting story here is that Banting has now started to recruit more patients, or the Toronto hospital. This is Teddy Ryder before and after the treatment. And this is Teddy Ryder at 72 years of age when he eventually died of other complications, and says, I shall always follow your career to Banting with interest, and you will forgive me if I add a little pride, because I shall always remember the difficult times we had in the early days of insulin. And Teddy Ryder and Banting had all these nice communications that if you ever go to Toronto, you will see all of this is in a museum. It's a wonderful experience to see all of this. I have not had the opportunity yet, but I definitely will at some point. And the interesting story here is that Banting was presenting this in a lecture, and there was a an executive from Eli Lilly, the company that you all know is this huge company with a lot of revenue now. So the executive was sitting and wanted to collaborate with Banting, and Banting, of course, refused. And then they met together again, and then it turned out to be one of the biggest collaborations ever. Eli Lilly is now one of the biggest manufacturers of insulin, and this is in the Toronto Daily Star. And this news, as I said, spread like wildfire, and the rest is history. Eli Lilly started making a ton of uh, insulin from the this led to the Nobel Prize, and this is till date. I was surprised to find this out, is that till date this is the earliest from discovery to the Nobel Prize. It just took I think less than 2 years for Banting and MacLeod to get the Nobel Prize. Banting thought that it wasn't MacLeod who deserved this prize with him, but Best. But that's another story. And he gave half of his prize money to Charles Best, and MacLeod gave half of his prize money to Collip. So in the end, it was sort of a happy ending. But what John Williams, the chief of medicine, has said, and many people have said, talked, and written about this, few recoveries from impending death have been more dramatic than this and ever witnessed by a physician. And the story goes that when they had, for example, 6 patients, all of them in a diabetic coma, they started injecting them with the insulin that they had purified from cows and pigs. The story goes that by the time the 6th kid was being injected, the first kid was waking up from the coma, and the parents were saying that this is nothing short of a miracle. So you can see how big this discovery was. And the you needed when you make it from pigs or from cows, as Eli Lilly was doing, you need 8,000 pounds or 3,600 kilograms of pancreas glands, which is roughly from 23,000 animals, to make 1 pound or half a kilogram of insulin. So you can see that even though this problem was solved, it took an enormous amount of effort. Eli Lilly went to all the slaughterhouses in and around Toronto, and not Toronto, sorry, all across the United States and later the world, to actually source their insulin. But this is where the insulin come from. In a separate story, I'll tell you how this was changed completely due to advances in genetic engineering. But that's a story for another day. So I have talked about all the people from the ancient world, Sushruth, Ibn Sina, and then Claude Bernard, the Europeans, and the North Americans. But you notice how all of these scientists, and uh, I also talked about how the scene of action shifted from Asia to Europe, and then to North America. And this is, of course, possible because of very curious people. But you notice how all of these are men. All of these scientists are men. And the reason, of course, is that women played a different role at that time in society. and this was considered the norm. And it is now that you see that increasing number of women are actually participating. And so the big challenge that remained was, how do you detect insulin in the blood? So Rosalind Yalow discovered this assay called the radio immunoassay, which is a very highly sensitive method to detect insulin, and she won the Nobel Prize for this effort. When she was interviewed, she said that the only difference between men and women in science is that women have the babies. This makes it more difficult for women in science, but it's merely another challenge to be overcome. Really astounding, a very incredible woman. And you will see that as you now know, she won the Nobel Prize here. As you now know, there is an increasing number of women now in the workforce that are doing extremely good science, but also, as the last few years have shown, making discoveries that lead to Nobel Prizes that will one day change the world, and already changing the world. This is Jennifer Doudna. The uh, story of CRISPR will have to be told in a separate session, one of the most phenomenal stories of our time, and also the story of Kathleen Caitlin Carrico, who won the Nobel Prize recently for her discovery that led to the vaccines that saved us from COVID-19. So this is again just to make the point that we have half of humanity has not participated sufficiently in science, which is now changing. And the other reason I bring this up is that there is one more story here that I will tell you one day, is that the one of the big reasons why this is possible is the discovery of another hormone and its effects, estrogen. And the story is absolutely fascinating. And I will recount that in a later episode, because there are many interesting facets to that. So, almost exactly 100 years ago, a dog, Marjorie, was injected with insulin. This changed, as I said, the world. It is saving now millions of lives worldwide. I was reading there's tens of millions of people who are alive just because of Banting and all the other people preceding Banting's efforts to save to find insulin and how it can reverse type 1 diabetes. This is one interesting thing. Dogs played a big role in the story, and now dogs are still playing a role, although it's a happier ending for dogs here. Dogs are being dogs are being trained to sense ketones or other substances in our body, where they can sense if the sugar goes too low. As Pam was mentioning, one of the big problems with type 1 individuals is that the sugar can really become so low that it's actually dangerous for them. So there are dogs that are being trained where they will sense this low sugar, and they will actually pack the patient, usually a kid, so that they know that their sugar is low. This is a very interesting story. Of course, does it work all the time? It is still old school science. It There was this interesting case where a patient actually sued the Diabetic Alert Dogs of America, the company that sells these very expensive trained dogs, saying that they don't work. They found that the dogs actually will sleep when the kid is sleeping, and that's why that's one of the reasons it doesn't work.
So,
Uh,
Ani, I'm sorry,
yeah,
might
no,
be
go
a
ahead.
good time to mention, parents of kids like Calvin have told me that ever since their child was diagnosed with type 1 diabetes and started taking insulin, they've never slept through the night. The challenge for parents and people who take insulin is they know they're supposed to keep their blood sugar from going too high, because too high over a long period of time leads to complications. But the danger is going too low, which immediately can send somebody into a coma. And that's why many parents have never slept again through the night, because they wake up in the night to check that their child's blood sugar isn't too low. And that's how these dogs that can sense that in the breath of the patient have helped many parents finally sleep.
That's wonderful. Thank you, Pam, for making that comment. What they noticed also is that diabetic individuals who have dogs generally do better, and that's also an interesting. I don't know how sound the science is, but
Right?
interestingly, you know that people who have pets and dogs, they have healthier lives, which is
Yeah.
another
Pet
topic for
therapy. Yeah.
and therapy. So now I'll end it by saying that, of course, this is an astounding story of a miraculous discovery, but there are big challenges that remain. And throughout the talk, Pam has been telling us what the challenges are. The insulin we have is not good enough. There are many other types of insulin that are being made. There are, of course, also glucose sensors that sense in real time how much glucose there is to deal with the problem that Pam really very nicely put of hypoglycemia or low blood sugar that can be more dangerous sometimes than high blood sugar. And the other thing is that we did not discuss today is the type 2 diabetes. Pam mentioned very nicely they are resistant to insulin, so you can have insulin, you can inject them, but in most patients it will not do anything because it's like someone knocking on your door, but nobody's there. So the number of type 2 diabetes patients are increasing throughout the world, especially in India. We can talk about when and why. I was talking to Anirban yesterday. There's a beautiful story here about not just what you do, but what your parents have done in the past and how it affects your diabetes risk that we can do in a separate episode. And also, the treatment of type 2 diabetes remains a big challenge. So, what we are going to talk about next is glucose monitors and these advances. There are some advances in stem cell therapies and others that I will be delighted if Pam joins us again for the next episode and offers her expertise to tell us about that. And what I'm really excited by is that I'm going to talk about if you have an elderly or if you're above 60 years of age or you have someone in your family, and this is especially true when I visit India and other places, is that there is next to their bed often a big pack of medications, and one of them is definitely the extract of this plant from Europe. And I'm going to tell you the fascinating story of metformin in the next episode. And what is even more fascinating, and this is sort of the talk of the town right now, is a magical substance. And of course, I'm using the word magical a little bit um, here. From this that of course the substance did not come from this lizard, but just knowing what is happening in a lizard in Arizona and in the southwestern United States, studying why this lizard actually fasts for a long time, finding what is in the venom of this lizard has led to a a substance that probably is one of the biggest blockbuster drug discoveries of the decade, if not more. And so we will talk about a drug that is up and coming, that is shaking the stock markets and leading to many people wanting to go into the black markets. And of course, we will tell you what it does and what it doesn't in a later episode. So I will end with that. And I just want to thank everyone who attended. And
So, Ani, we have one more question
okay.
before we wrap up. Which is more dangerous, broadly speaking, type 1 or type 2 diabetes?
Would you like me to address that?
Yes,
Sure,
I think.
sure,
Yeah.
Pam. Yeah.
So in the immediate diagnosis, type 1 is more dangerous. Without
Because
insulin, patients will die. Type 2 tends to be a little more gradual in what happens. And so there is some insulin being made and the cells maybe are taking up some sugar out of the bloodstream. It tends to get worse over time. So I would say immediately after diagnosis, in general, type 1, but that's not always the case because some people with type 2 might not get diagnosed till very late in the disease process, and at which point they need insulin as well. All right.
Well, I think that was last of the questions. We can wrap up, Ani. You're muted.
Okay, I apologize. We were a bit over time here, but thank you all for attending. And I hope that you will come back for the next story, especially the story of Ozempic and the lizard is going to be really, really
Yeah.
interesting.
So, diabetes story really set us for the Ozempic, which is the topic of the moment these days. And so, yeah, I think that's what I find so fascinating about science. Like, looks like an impossible discovery, but if you go back, it's really progressive in a step by step. Like first observing the symptoms, sweet urine, then finding which organ it is, and then finding which cells it is, and then okay, how can we get more of that? So you unfortunately kill some, butcher some animals, get that, and then we say, okay, drug companies come in. Okay, how can we do that synthetically, make that in the lab? And so this is how the progression is, and some people get the Nobel Prize, but so many people have made small contributions without which the Nobel Prize would never have happened. So I think we should also be appreciative of all those scientists which made tiny contribution, took the field one step forward, and someone came in the end and got the Nobel Prize. So it's a huge collaborative effort between so many different people from different expertise areas.
That's so true. Yes.
Absolutely.
All
All
right.
right. Thank you. And I hope you all come back.
Awesome.
Thank
Thank
you.
you.
Thank you.
Bye.